Yep. All right, before we begin, are there any questions? Can somebody please confirm you can hear me?
Yes, we can hear you.
Great.
I have a quick question.
Yeah, go ahead and ask.
So, about the unknowns project, we haven't received any email about our numbers of bacteria, so I was just wondering when we'll get it.
Hopefully tonight.
Hopefully tonight. Okay, thank you.
All right, give me a minute and I'll be right with you. All right, let me share my screen. Let's go to the syllabus. With the unknown project, I'm a little behind schedule. I've still got to get the information together, and explained in my 3 o'clock lab that I've got to figure out how to send it to the 3 p.m. lab and the 7 p.m. lab because everybody's together. And if I can't figure it out, you're going to get 2 documents. One will be for the 3 p.m. and one for the 7 p.m. lab. All right, let me share my screen here. And today is February 13th. And we're supposed to be on chapter 13, chapter 5, and finishing chapter 5, and hopefully starting a little bit of chapter 6. Any question about any of that? Do note that I postponed the quiz. Quiz 3 will not be held this Wednesday. It'll be a week from this Wednesday. And that forced me also to move quiz 4, postponing it a week as well. And I need to upload the syllabus. You don't have the new syllabus with the change in the schedule. I made an announcement about that. The reason was because a student had pointed out that on quiz 3, which is going to cover lab 6, and I don't have that graded for the first time yet. And there's just no way I'm going to get that graded. If you haven't noticed, I'm in the middle of grading lab 5. I've got about a third of the students graded on lab 5 the first time, and that's all I've got. I'm going to work on that tonight and hopefully get the rest of you graded tonight in lab 5. But to get lab 6 graded tonight, it's just going to be too much. And so I had to change the schedule because I don't want you to have quiz questions that you don't know may have been answered wrong in your lab. All right. I can't get that to move up. Let's skip it. let's go to presentation. And this is the slide we had talked about. I believe I've shown you the movie on aerobic respiration. And I told you that to produce energy from glucose, microorganisms use two general processes, respiration, and that can be aerobic respiration, which the video showed you, and anaerobic respiration. And the other process is fermentation. Is that correct? Anyone?
Yes, that's correct.
Thank you. Wait a minute. Let me go ahead and remove the Tuesday, Thursday. Save that down. Surprising, I'm staying together on Monday, Wednesday, and Tuesday, Thursday, although Monday, Wednesday is a day behind schedule because one day we did not have class because the internet was down. Not the internet all over the place, just the connection to Clark College. So I couldn't log in and nobody else could either. All right, we're looking at this slide here showing you aerobic respiration in general compared to fermentation. Aerobic respiration has three principal stages: glycolysis, the preparatory step in the Krebs cycle, excuse me, and then the third step of aerobic respiration, the electron transport chain, also abbreviated ETC. Essentially, electrons are going to flow from glucose, which is an energy-rich molecule, to CO2 and water, energy-poor molecules, and we'll get a high yield of ATP from aerobic respiration. Compared to fermentation, shown on the right here. Oh, no, that's in the way. I can't get, there it goes. It does involve glycolysis. That's the start of fermentation and the start of aerobic respiration too. There is no Krebs cycle. There's no electron transport chain. Instead, we have one or two fermentation steps after glycolysis where pyruvate is converted into
So when he signed or did all this, he did it so everybody could be on mute. So you have to physically come off to.
Okay, somebody's talking and you're not muted. Could you mute your microphone, please? Let me see if I can figure out who that was. Didn't I stop my share? No, I didn't. All right, they're now muted. So, yeah, after you talk, you want to mute yourself unless you're not going to make any noise. Let's share my screen again. So in fermentation, we have a fermentation step where the pyruvate of glycolysis is converted into the end product of fermentation. That can be ethanol for alcoholic fermentation, or can be lactic acid for lactic acid fermentation. It can be another end product depending on the fermentation step. There is a much lower ATP yield from fermentation than aerobic respiration. Come on. In aerobic respiration, once again, this slide is showing you the overview. So if this pink box here is the cell, then glycolysis is occurring in the cytoplasm of the cell, where glucose is converted into pyruvate. There is some ATP that's made, some NADH that's made, and then the pyruvate in a eukaryote cell will cross into the mitochondria, and in the matrix of the mitochondria will engage in the Krebs cycle, also called the citric acid cycle, where there'll be some ATP that's made, some of the pyruvate will be converted into CO2, and actually in the end of the Krebs cycle, all of it will be converted to CO2. And some NADH and FADH2 are made. The NADH and the FADH2 carry the electrons from ultimately glucose to the third step of the electron transport chain, which is the electron transport chain, also called oxidative phosphorylation, where we produce ATP in high numbers. And this happens, the electron transport chain, in and across the inner mitochondrial membrane. So this is trying to show you where the aerobic respiration is occurring in a eukaryote cell. Now, prokaryotes do not have a mitochondria. Glycolysis happens the same. I think I have that shown in the next slide, but let me take a look here. No, I don't. It's going to be shown in another place. In prokaryotes, we have glycolysis happening in the cytoplasm. The Krebs cycle also happens in the cytoplasm because there is no mitochondria. And then the electron transport chain does not happen across the inner mitochondrial membrane. It happens across the cell membrane. So the electron transport chain in prokaryotes happens across the cell membrane. Any question about any of that? All right. In aerobic respiration, the first step is glycolysis. That does have another name. I don't use this name, the Emden-Meyerhof pathway, and I could be butchering their names because I'm not German or German speaking. And they're the two scientists who discovered the glycolysis pathway. In glycolysis, we have the oxidation of glucose. Glucose is converted into pyruvic acid. There are two pyruvic acids made because this is a 3-carbon molecule and glucose is a 6-carbon molecule. Glycolysis occurs in most living cells. It can occur in the presence or the absence of oxygen. It does produce some ATP and some NADH, but not a lot. And there we're seeing the steps of respiration here. Glycolysis is shown here where it, let me blow this up. Starts with glucose on the top. Let's go down there. And it's converted to this intermediate, converted to this intermediate, converted to this intermediate, but takes in 2 ATPs. So 2 ATPs are used. You make this intermediate and that intermediate. This intermediate can convert into that intermediate. And then this intermediate can convert down this step, ending in pyruvic acid. You can see that 2 ATP come out here and 2 there, so that in glycolysis we get 4 ATP that are generated, but 2 ATP come in, so there's a net gain of 2 ATP. Any question about any of that? Another thing I need to mention, and that is sometimes people will call this pyruvic acid, what ends up from glycolysis. Other times they'll call it pyruvate. The only difference is whether the hydrogen atom is attached or not, and the cell will treat pyruvate and pyruvic acid the same. And you need really a chemist to distinguish between the two. So we will use those two words interchangeably, although the chemist would not like that, but the cell does use them interchangeably. Any question about any of that? Now, when I was an undergraduate, I had to learn all of the intermediates of glycolysis and all of the enzymes performing each of the steps in the biochemical reactions of glycolysis. I think it's fair that because I had to know that, that I require you guys to know that. What do you guys say about that?
Hilarious.
Delirious. Hilarious, whatever. You don't need to know that, but you do need to know the summary reaction. You need to know the reaction starts with glucose. It ends up with 2 ATP, which is a net gain of 2 ATP. You get 2 pyruvates, also called pyruvic acid, and you get 2 NADH, which, let me see if I can find them here. There's one there. Actually, there's 2 there. So that's the summary reaction, where you start with glucose and you end up with 2 pyruvic acids, a net gain of 2 ATP, and 2 NADH. Any questions about glycolysis? All right. Before we move further, let me state that respiration is the oxidation of molecules, releasing electrons for the electron transport chain. There are 2 forms of respiration: aerobic respiration, where the final electron acceptor is oxygen, meaning that the electrons that start from glucose flow through different molecules and they end up at oxygen. So we say oxygen is the final electron acceptor. With anaerobic respiration, we have the same reactions occurring, but the oxygen does not flow to, the electrons do not flow to oxygen. They flow to some other molecule other than oxygen. The exact molecule the electrons flow to depend on which form of anaerobic respiration we're talking about. There are different forms, like there's nitrite anaerobic respiration, where the electrons flow to nitrite. There's nitrate anaerobic respiration, where the electrons flow to nitrate. You could have iron respiration, where the electrons flow to iron. There's many different forms of anaerobic respiration, and the electrons end up at another molecule. All you need to know is that molecule is not oxygen. ATP is generated in all forms of respiration, both aerobic and anaerobic. The amounts of ATP differ depending on which form of respiration we're talking about. And both aerobic respiration and anaerobic respiration have the glycolysis. They have the preparatory step in the Krebs cycle, and then they have an electron transport chain. It's the electron transport chain which differs between aerobic respiration and anaerobic respiration. And we'll look a little bit more at that when we talk about anaerobic respiration. But at the moment, we need to talk about aerobic respiration. Any questions about that? All right, so in aerobic respiration, we have glycolysis, and then we have the preparatory step and the Krebs cycle. The preparatory step is essentially one reaction where the pyruvic acid is being prepared for entry into the Krebs cycle. Pyruvic acid loses 1 carbon, so one carbon is burned off of pyruvic acid as CO2. Coenzyme A comes in and binds to the remainder of pyruvic acid, and that becomes acetyl coenzyme A. In the process, NADH is generated. And now the acetyl coenzyme A can directly enter the Krebs cycle. There will be 2 molecules of acetyl coenzyme A made per molecule of glucose. So the preparatory step will run twice for each molecule of glucose, because glucose will be made into 2 pyruvic acid molecules. Any question about that? All right. So after the preparatory step, we have the Krebs cycle. The Krebs cycle has a number of different names. I and your textbook author tend to use the term Krebs cycle after the doctor who discovered the Krebs cycle or the cycle. And it's also called tricarboxylic acid cycle, or an abbreviation TCA, because of the number of tricarboxylic acids in it. It's also called the citric acid cycle, and that's also abbreviated T for the, C for citric, and A for acid, so TCA works for both. And that's because the first molecule made in the Krebs cycle is citric acid. So there we're seeing the preparatory step on the top, converting the pyruvic acid to acetyl coenzyme A. Acetyl coenzyme A then enters the Krebs cycle and will bind to oxaloacetic acid, making citric acid. You don't need to know really citric acid because you don't need to know any of the intermediates except for the last one, and that is citric acid is made into this one, which is made into this intermediate, made into that intermediate, made into that intermediate. And you can see some ATP is actually made on that step, made into that intermediate, made into that intermediate, which then generates the molecule that we started the citric acid cycle, oxaloacetic acid. And that's why it's called the cycle. This reaction runs in a circle and ends up at the place it begins. So as long as you're alive, the citric acid cycle or the Krebs cycle will be running, assuming you have something to eat and put into it, assuming you're not starving. You should be able to get a molecule to put in and then get the Krebs cycle to run. And it'll just keep running for as long as you're alive. In the Krebs cycle, you make ATP. There's one right there. You make 4 NADHs. The first is in the preparatory step. There's another one there, and the fourth one there. We make 1 FADH2 molecule, that's there. And NADH and FADH2 carry the electrons from, ultimately, the electrons came from glucose, and they will be carried to the electron transport chain. We also make 3 CO2 from the preparatory step in the Krebs cycle. The first one's in the preparatory step. The second one's right here, and the third one is right here. Now remember that 2 pyruvic acids are made by glycolysis for each molecule of glucose. So both the preparatory step and the Krebs cycle will run 2 times for each molecule of glucose. That means of the 6 carbons of glucose, all 6 of them will be converted into CO2. So 2 here, 2 there, and then 2 there. Okay? All right, any question about the preparatory step and the Krebs cycle?
I'm sorry, I might have missed this, but did you say the preparatory step was the same thing as glycolysis?
No, it's
No.
the step after glycolysis.
Okay,
The
okay.
main steps of aerobic respiration are 1, glycolysis, 2, the preparatory step in the Krebs cycle. And I'm linking them together because the preparatory step is the start of the Krebs cycle.
Okay, got it. Thank you.
All right.
Great
all right.
question.
All right, so you need to know the box in red. This is the amount of ATP, NADH, and FADH2 and CO2 you generate from one cycle of the preparatory step in the Krebs cycle. But you should remember that you run this cycle twice. So if I'm saying how many ATP do you make in the preparatory step in the Krebs cycle from glucose, you'd have to multiply by 2 to get that answer. Any question about that? All right. So there we're looking at glycolysis and then the preparatory step in the Krebs cycle. And we make some NADH and some FADH2, which carry the electrons that ultimately came from glucose. And those electrons are carried by NADH and FADH2 to the electron transport chain. In the electron transport chain, we make a high number of ATP molecules. So far, we've only made 4, a net gain of 2 in glycolysis and a net gain of 2 in the preparatory step in the Krebs cycle. Let me end the show here. So the electron transport chain is a series of carrier molecules that are in turn reduced and oxidized as the electrons, which are flowing from glucose, are passed down the chain, meaning NADH or FADH2 bring the electrons to an early member of the electron transport chain. NADH2 brings it to the first member of the electron transport chain. And then the electron moves to the second member, it moves to the third member, moves to the fourth member, etc. Energy is released as electrons are transferred from a high energy molecule, such as glucose, to the lower energy compounds, which would be CO2 and water. This energy is released and used by chemoosmosis to produce ATP. Let's watch a little video on the electron transport chain.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, 2 mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
All right, any questions about the video? No, let's close that down. So in eukaryotes, the electron transport chain occurs along the membrane of the inner mitochondrial membrane. In prokaryotes, it occurs along the cell membrane. Let me blow this up. When do I want to talk about that? I guess I'll talk about it here. And that is in the mitochondria, in the electron transport chain, we pump the hydrogen ions from this side of the membrane, which is inside the inner mitochondrial membrane. So this is the matrix, to outside. And then we have the outer mitochondrial membrane. So the hydrogen ions stay between the inner membrane and the outer membrane. And that helps protect the cell. These hydrogen ions don't leave the mitochondria. Now, in the prokaryote, this is the cell membrane. And where the hydrogen ions are pumped is from the cytoplasm inside the cell to where the cell wall is, assuming the bacteria has a cell wall. Any question about any of that? All right. So in the electron transport chain, NADH carries the electrons to the first protein in a membrane. And this is why it's called a electron transport chain. The electrons transport from the first protein in the membrane to the second, come on mouse, second protein in the membrane, and then from the second protein in the membrane to the third, to the fourth, to the fifth. And then the electrons leave the electron transport chain, and in aerobic respiration, the electrons flow to oxygen. So oxygen is the final electron acceptor. The electrons and hydrogen ions combine with oxygen to produce water. And that is why we have to breathe oxygen in. All the oxygen we breathe in is converted in the electron transport chain to water. And that water, we'll breathe some of it out, and some of it will be used by us as water for us, meaning we generate water in aerobic respiration. Desert animals are actually very good at conserving this water that they derive from aerobic respiration, like the desert mouse, and they get 65% of the water they need from this reaction of aerobic respiration. Breathing in oxygen, combining it with electrons and hydrogen ions to make water. Any question about any of that? All right. As I mentioned, NADH, go on. NADH delivers the electrons to the first protein in the membrane. Why it's called the electron transport chain. It's a chain of proteins in the membrane, and the electrons transport from one protein in the chain to the next. When the electrons move through certain of the proteins, that includes the first one, come on, mouse, right there, the third one right there, and then the fifth one right here. When the electron is moving through these proteins, it gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. That creates a twofold gradient, different, and that is there's more hydrogen on this side of the membrane than that side. There's also more positive charges on this side of the membrane than that side. That makes potential energy. These hydrogen ions will want to move from this side of the membrane to that side of the membrane, but they can only move across the lipid bilayer at one place, and that's this protein. And this protein is a ATP synthase molecule. When hydrogen ions move through it, each hydrogen ion gives this protein the energy it needs to convert ADP, an inorganic phosphate, into ATP. And that is how the cell makes ATP. By pumping the hydrogen ions on this side, creating a twofold gradient, meaning there's potential energy, there's more hydrogen on this side than this side, so the hydrogen want to move here. There's more positive charges on this side of the membrane than that side, so the positive charges want to move this way. This potential energy is used to make ATP by this protein here, which this is an ATP synthase enzyme. Any question about any of that?
Where does the inorganic phosphate come from?
It's phosphate in the cell. Some of it will come from when ATP is broken down into ADP and phosphate, and then it's just recycled. So it's used to generate the ATP by combining the phosphate with the ADP. Green plants can absorb phosphorus from rocks or whatnot, and they can also take phosphate in a fertilizer. You'll note that probably the second most common chemical element in a man-made fertilizer, meaning where they're putting in the chemicals, is going to be phosphate, some molecule containing phosphate. The number one will, of course, be nitrate, because these are the two elements that green plants need and want, and so they will be in fertilizer. Now, humans, we can get phosphate from drinking milk, obviously breaking down ATP. We might be able to get phosphate also from breaking down some proteins. But we need phosphate. We can't easily make phosphate, where green plants can take any form of phosphorus and make phosphate. Is that a good enough answer?
Yeah, that's great. Thank you.
Okay.
Can you repeat where the electron transport chain occurs for each of the cell types? So for eukaryotes, it's along the cell membrane, right?
No, for prokaryotes, it's along the cell membrane.
Ah,
This membrane here is in a prokaryote, the cell membrane. This is inside the cell, the cytoplasm. That is outside the cell. And for most prokaryotes, that'll be in the cell wall, meaning they pump the hydrogen ions in the cell wall. And in eukaryotes, this is the inner mitochondrial membrane. So this will be the matrix of the mitochondria. This will be the space between the inner membrane and the outer membrane of the mitochondria.
thank you.
Okay. FADH2 is not as efficient as NADH. FADH2 brings the electrons to the second protein in the electron transport chain, and that misses the first protein, which can pump hydrogen ions across the membrane. So FADH2 is not as efficient at generating ATP as NADH is. Any question about any of that? All right, let's move on. So ATP molecules produced by the electron transport chain of aerobic respiration. In eukaryote cells, we produce 32 to 34 molecules of ATP per molecule of glucose. Prokaryotes are a little more efficient. They always generate 34 molecules of ATP per molecule of glucose. It's curious in the eukaryotes that most of our cells generate 32 molecules of ATP per molecule of glucose. So that would be our skin cells, our nose cells, our muscle cells, our brain cells. But there are 3 cells which are more efficient, and they generate 34 molecules of ATP per molecule of glucose. And those cells are the heart cells, the liver cells, and the kidney cells. They generate 34 molecules of ATP per molecule of glucose. What do you know about the heart, which implies that it would probably be more efficient than your skin cells or your muscle cells? Since nobody's giving an answer, I'll say, or your brain cells. Why is the brain cell less efficient than the heart? What do you know about the heart? While you're alive, what's the heart always doing?
Pumping.
Yeah, the pump, the heart, the kidneys, and the liver are always working. Those organs are always working. That's not the case for your skin cells, or your muscle cells, or even your brain cells. They do not work all the time. So the heart, the kidneys, and the liver are more efficient at generating ATP because those cells are constantly working. They are constantly using up ATP, meaning to get the heart to beat, the heart has to use ATP. So those cells are more efficient, and they always generate 34 molecules of ATP per molecule of glucose. All the other cells in your body, which are most of them, are not as efficient, and they only generate 32 molecules of ATP. The final electron acceptor in aerobic respiration is a molecule of oxygen. Oxygen will combine with hydrogen to form water. And as I stated, let me go back just a minute. All of the oxygen you breathe is not converted into CO2. All of it is converted into water in the electron transport chain. All of the CO2 you breathe out comes from the burning of glucose. Each of the atoms of glucose is converted into a molecule of CO2. Any question about any of that? So this is showing you the electron transport chain right here. This is the overview of aerobic respiration compared to fermentation, and that's showing you where it's happening in a cell. So the electrons flow from glucose into NADH or FADH2. These molecules bring the electrons to the electron transport chain, and then the electron moves from one member of the electron transport chain to the next. And each step, the electron loses energy so that the energy is given off in slow steps so the cell can continue to be at 37 degrees Celsius, as opposed if you were to light the glucose, that would be like 1,000 degrees. I don't remember that's 1,000 degrees Fahrenheit or 1,000 degrees Celsius, but that's the temperature that you can burn glucose at when you take a blowtorch to it. And you don't release the energy in steps, and then the temperature is much higher. In aerobic respiration, that electron flows from one molecule in the electron transport chain, come on mouse, to here, and then flows to here, to there. And these are all, this one initially would gain the electron, so that is a reduction, and then it loses the electron, that would be oxidation. And then this one gains it. So these are all oxidation reduction reactions until the electron flows to oxygen, converting the oxygen into water. And this is showing you the location of electron transport chain in the cell membrane of a prokaryote cell, or the inner mitochondrial membrane of a eukaryote cell. Any questions on any of this? We talked about that, let's talk about this. Chemoosmosis is the making of ATP, where the hydrogen ions from this side of the membrane flow to this side through the ATP synthase, making ATP. That's what chemoosmosis is. It is a part of the electron transport chain. I do not use this terminology because as it is, or oxidative phosphorylation, all of those terms are used for the electron transport chain. And there's enough terms in science that I try not to use terms that are not similar, like the electron transport chain, which is the term I'm using, and the term that Totoro mostly uses, does mean the same thing as the electron transfer chain. Okay, the electron transport chain, the electron transfer chain. And those two names don't tend to foul students up. They're close enough that people don't get confused. But when we start throwing chemoosmosis or oxidative phosphorylation, those are different terms, and I just don't use them to keep things a little bit simpler. It's called oxidative phosphorylation because ADP is phosphorylated. That means it gains a phosphate group, and it's oxidative because the electrons are being lost. So in aerobic respiration, the complete oxidation of glucose occurs via aerobic respiration that yields between 36 to 38 ATP molecules in a eukaryote cell, depending on which eukaryote cell we're talking about. Most generate 36. In a prokaryote, they're a little more efficient, and they always generate 38 molecules of ATP for aerobic respiration. The overall equation for aerobic respiration is glucose, C6H12O6, plus 6 oxygen molecules, plus 38 ADP and 38 phosphates, generate 6 CO2 molecules, 6 water molecules, and 38 ATP molecules. Any question about any of that? You do need to know the overall equation for aerobic respiration. All right, so here we're looking at aerobic respiration in prokaryotes compared to, is that fermentation? Let me blow that up so I can see it better. No, this is just aerobic respiration generating NADH in glycolysis, the preparatory step in the Krebs cycle, the generation of ATP and CO2. We generate 2 ATP, a net gain in glycolysis, 2 in the preparatory step in the Krebs cycle. And then for prokaryotes, 34 ATP in the electron transport chain. In eukaryotes, it's either 32 or 34, for a total of 38 ATP in prokaryotes for aerobic respiration. Any question about any of this? Well, let's just move on. All right. So we've talked about aerobic respiration. Now let's talk about anaerobic respiration. Anaerobic respiration is similar to aerobic respiration. The main difference is that the final electron acceptor is different. It is not oxygen. It's an inorganic molecule that is not oxygen. It could be nitrite, could be nitrite, nitrate, nitrite. It could be sulfate. Nitrate is shown here. Nitrite would be NO2 with a negative charge. Sulfate, it could be iron. It just depends on which form of anaerobic respiration we're talking about. In anaerobic respiration, glycolysis and the Krebs cycle still occur. You do have an electron transport chain in anaerobic respiration. However, this electron transport chain is shorter than in aerobic respiration. It depends on which form of anaerobic respiration we're referring to, but the yield of ATP in anaerobic respiration is less than the yield in aerobic respiration. The yield is always greater than 2 ATP for anaerobic respiration and up to 36. So the best form of anaerobic respiration generates 36 ATP. The worst is about 4 molecules of ATP. And it depends on which form of anaerobic respiration we're talking about for how many ATP molecules we make. Let me go back to the slide showing you the electron transport chain. Here we go. Of course, that would be better here. The electron transport chain is a little smaller in anaerobic respiration. Some of them lack the first protein. Others lack, I think they lack everything except for the last protein. You don't need to know this, but like I said, the electron transport chain is a little smaller in the anaerobic forms of respiration. And you make less ATP in the process. Depending on which form of anaerobic respiration we're talking about, there's many different forms of anaerobic respiration, and we're not going to go through each of them. Any question about any of that? All right, because anaerobes, meaning organisms that perform anaerobic respiration, make less ATP than aerobes, organisms that perform aerobic respiration, the anaerobes tend to grow slower than the aerobes. Any question about that? All right, let's talk about fermentation. Here we're looking at fermentation on the right side and respiration, mostly aerobic respiration on the right side. And we know it's aerobic because there's oxygen there, but it could be any form of respiration on the left side. Fermentation does start with glycolysis, the same as aerobic respiration. We do generate some ATP from glycolysis. We do generate pyruvic acid. That pyruvic acid is changed in 1 or 2 fermentation steps, and then we make the fermentation end product. So fermentation is much simpler than aerobic respiration. We have glycolysis, so that's the same. But then in only 1 or 2 steps, we make the fermentation end products. It depends on which fermentation we're talking about, how many steps it has. We're only going to talk about 2 types of fermentation, alcoholic fermentation and lactic acid fermentation, but Drew realized there's a handful of different fermentation reactions. Many fewer than forms of anaerobic respiration. Any question about any of that? All right, fermentation releases energy from oxidation of an organic molecule via glycolysis. Usually glucose is used, and glucose will be oxidized to make pyruvate. Glycolysis does proceed in fermentation as discussed previously, so we get a net gain of glycolysis of 2 net ATP, some 1 NADH, and then we generate pyruvate from the glucose. Now understand, glucose is the preferred molecule, but you may ferment other sugars, other amino acids, other molecules. Most of these other molecules will be converted to glucose, and then glucose will be sent through fermentation. So let's talk about pyruvic acid fermentation. Glycolysis makes pyruvate. We do generate 2 NADH by glycolysis and 2 ATP. That 2 ATP is all the ATP we get out of fermentation from glucose. The pyruvic acid is converted in one fermentation step. So this one's only one step into lactic acid, the end product of lactic acid fermentation. And you'll note that in the fermentation step, we use the NADH to make NAD+. My question for you, let's see how much time we have. We do have enough time, I think. My question for you is, why does the cell use this NADH up? Like when our muscle cells are working very hard and we start running out of oxygen, so there's not enough oxygen around to perform aerobic respiration, the muscle cells don't just stop. They will engage in fermentation to make ATP so that the muscles can continue working. Once you have no ATP production, the muscles cannot work. And so our muscles will initially use aerobic respiration, but if they're working at a extreme rate or something that's very physical, meaning very taxing on the muscle will use up all of the oxygen in aerobic respiration. And then aerobic respiration cannot perform because there's no oxygen around. And so the muscle will fall back on fermentation, specifically lactic acid fermentation shown here, where glucose is converted to pyruvic acid. Pyruvic acid is converted into lactate. Let me go one more right there, shown on the left. Why does your muscle cell use up NADH in the fermentation step rather than just keep it around for aerobic respiration? Because your hearts and your lung are working like mad to bring oxygen and blood to the muscles, but the muscle is using up the oxygen faster than it can be brought in, so the muscle falls back on fermentation. My question is, why doesn't the cell just retain the NADH and wait until the oxygen returns to the cell and then use that NADH to make ATP in aerobic respiration? Because you'd make a lot more ATP that way than you do in fermentation. Anyone have a guess? It's actually shown in the slide here. Nobody's going to guess? Well, the muscle cell needs to use the NADH for fermentation to happen. Otherwise, there won't be NAD plus around to have glycolysis happen. And if there's no NAD plus around, glycolysis can't happen. And if the glycolysis isn't going to happen, we won't make any ATP. the muscle cell wants to make some ATP so the muscle can continue working. And so that's why the cell recycles this NADH to make NAD plus so that the glycolysis can continue working. If you've ever seen an Olympic athlete running in the Olympics or doing something in the Olympics, and then they just collapse, it's because they actually ran out of glucose to work the muscle. And without glucose, fermentation cannot happen. Without glucose and oxygen, aerobic respiration cannot happen. And as I mentioned, the muscle will use up all of the oxygen. And so that's why the muscle falls back on fermentation. But when the muscle runs out of glucose, the muscle cannot make ATP, and then the muscles just relax. The Olympic athlete will fall down. In the summertime, the coach, I think, runs out and sprays the athlete with water. And in the wintertime, they run out and throw a blanket over the player. And then the player gets up because the hearts and the lungs are working like mad to send the muscle oxygen in the blood as well as glucose in the blood. And once the muscle cell gets sort of refed by the blood, the Olympic athlete can then stand up, and usually they walk across the finish line at that point. So they're quite fine. It's just that the muscles could no longer work, and then they just collapse. I'm sure you've seen that because almost every Olympics, we have one athlete who pushes themselves to that point where they use up all the glucose and then their muscles can't work. Anyways, that's lactic acid fermentation. Alcoholic fermentation is similar. The difference is we have two fermentation steps. We take pyruvic acid, ferment it to this intermediate, and then we take that intermediate and make ethanol. Why it's called alcoholic fermentation. It's alcoholic fermentation. We make ethanol as the end product. This happens in yeast cells where they can ferment glucose to alcohol. And we use these yeasts to leaven bread as well as other yeasts to make beer and wine. Any question about any of that? Now, there are different fermentation end products because there are different fermentation reactions. The two most common are lactic acid fermentation. Our muscle cells can do that. Lactobacillus that makes yogurt can do that. Almost all fermented foods, most cheeses like cheddar cheese, use lactic acid fermentation. Sauerkraut uses mostly this. I guess it uses this partly, but it does use other fermentation reactions too. Hopefully I didn't say bread does that. Bread uses a yeast, Saccharomyces, to make ethanol and CO2. We can use this yeast also to make beer and wine, to make alcohol. Those are the two. There are like 4 other fermentation reactions. The only one I'm going to talk about is propion bacterium, making propionic acid. This is actually the fermentation process that is used to make Swiss cheese. And that's why Swiss cheese tastes different than most other cheeses, because all other cheeses use lactic acid fermentation, except for Swiss cheese, which uses propionic acid fermentation. And propionic acid produces CO2, shown here. And when you look at Swiss cheese, you'll see a hole in it. That hole is being produced from the CO2 made in the fermentation step. Lactic acid fermentation does not produce any CO2. Alcoholic fermentation does make CO2. When we use yeast to make leavened bread, it's the CO2 that rises to make the bread leavened, meaning you get the bread having air pockets in it, making the bread dough rise. And we use this to make beer. The CO2 actually gives beer its fizz. In wine, we use an open casket or an open barrel, so the CO2 vents to the air. But if we were to make the wine in a bottle that is capped, so that we trap the CO2 in it, we call that champagne. Champagne is the same as wine, it's just you're trapping the CO2 in it. And that's why champagne is bubbly and wine is not. Any question about any of those? The other fermentation reactions are important in industry to make different products like lactic acid, succinic acid. This one for making butanol or acetone. This is showing you some of the different fermentation reactions, the industrial commercial use, the meaning of purpose, like ethanol fermentation using to make beer, wine, and it's not there, but that would be also leavened bread. Lactic acid fermentation to make most cheeses, yogurt, rye bread, sauerkraut, summer sausage, chocolate. And then the starting material, which you don't need to know. And then the microorganism performing the fermentation reaction. Any question about any of that? All right, let's look at aerobic respiration, anaerobic respiration, and fermentation, comparing the three. Aerobic respiration requires oxygen, that's why it's aerobic. Anaerobic respiration does not require oxygen. Oftentimes it requires an anaerobic growth condition. Fermentation can happen either in aerobic conditions or anaerobic condition. It does not use oxygen, but oxygen does not hinder fermentation. With many forms of anaerobic respiration, if you have oxygen around, anaerobic respiration cannot proceed. Many anaerobes are actually killed by oxygen. The final electron acceptor for aerobic respiration is oxygen. For anaerobic respiration, it's some inorganic substance other than oxygen. For fermentation, it is an organic molecule. So the pyruvate will have the electron flow to lactic acid in lactic acid fermentation. Actually, lactic acid is what ends up. Anyways, the type of phosphate you don't need to know about. And then the molecules of ATP being produced, 36 to 38 for eukaryotes, 38 for prokaryotes for aerobic respiration. For anaerobic respiration, it's variable, less than 38, so let's say 36, but more than 2, so let's say 4 to 36. And then fermentation, we only get 2 per molecule of glucose. Any question about any of that? All right, so glucose is the preferred molecule for performing all of these reactions. That includes fermentation, aerobic respiration, or anaerobic respiration, but any carbohydrate can be used. Usually you break down the carbohydrate and break it into glucose. Even if you were to eat a bunch of fructose, that molecule will be slowly converted by your liver into glucose and then released slowly into the blood as glucose. My point being that carbohydrates are generally broken down into glucose or converted into glucose. Another example would be lactose. You break it down into beta-galactose and glucose. The glucose will then enter aerobic respiration the normal way. The beta galactose has to be converted into glucose and then will enter the aerobic respiration. A problem with large carbohydrates and other large molecules that we're going to use for lipid or protein catabolism is that these large molecules cannot cross the cell membrane. However, lipids and proteins, as well as carbohydrates, can be used in a catabolic reaction to perform aerobic respiration or fermentation. As I mentioned, large molecules have a problem, and that is they cannot enter the cell because they can't get through the cell membrane. We have a way around this, and so do microbes, and that is they make lipases and proteases and then secrete them external to the cell. The same thing in our gut. Our gut cells will make lipases and proteases and then secrete it into the lumen of the gut. And then you break down the large molecules, the lipids and the proteins, into smaller monomers. And then the monomers can be brought into the cell membrane with a transport protein. Glycerol can be catabolized by glycolysis. The fatty acids of a lipid will be cleaved into 2 carbon chunks, and then those will enter the Krebs cycle. Amino acids will have the amine group removed. That's the NH2 group. And you'll usually pee that out. And then the remaining part of the molecule is converted. It usually enters the Krebs cycle, but it depends on which amino acid we're talking about. Any question about any of that? The point is that lipids, proteases, and complex carbohydrates can be digested and used in anaerobic respiration or aerobic respiration or fermentation. Most of the time, when we do that, for those of us couch potatoes who don't get a bunch of exercise in, we will convert these molecules into glucose and then send glucose down the aerobic respiration in the normal way. And this is trying to show you this slide, the interrelationship of carbohydrate, lipid, and protein catabolism. Proteins can be broken down into amino acids. Some of the amino acids will enter the glycolysis late in the process. Some amino acids will be converted into acetyl coenzyme A and then enter the first step of the Krebs cycle. Some amino acids will be converted into a later intermediate of the Krebs cycle and then enter the Krebs cycle lower down, not at the start. Lipids will be converted into glycerol, which will enter the glycolysis about midway through. The lipids will also be broken down into fatty acids. As I mentioned, they will be broken down into 2 carbon chunks, converted into acetyl coenzyme A, and then go through aerobic respiration the normal way. Carbohydrates are generally converted into sugars, which will then be converted into glucose, and then they enter aerobic respiration or anaerobic respiration or fermentation in the normal way. Any question about any of that? See what we're doing on the time. We've got 5 minutes. The complete metabolic oxidation of glucose in aerobic respiration is the most efficient, meaning aerobic respiration is the most efficient way to make ATP. The ATP is then used by the cell for a cellular process. This includes if the cell is moving, it's using ATP. If the cell is transporting a molecule across the membrane against its concentration gradient, it will use up ATP. If the molecule is synthesizing a new molecule, it'll need energy, and it will use that energy from breaking down ATP. So most cellular ATP is actually used in the last, and that's the synthesis of a new cellular component where anabolism occurs, where smaller molecules are made into a larger molecule, such as the biosynthesis of a protein. The metabolism of the different foods or food molecules like proteins, lipids, and carbohydrates is integrated, and the catabolic pathways are joined by common intermediates. We call a pathway that can function in both directions an amphibolic pathway. Amphibolic pathways bridge the pathways involved in carbohydrate, lipid, protein, and nucleic acid metabolism. Here we can see, let me see if I can blow this up a little, that wherever we have two arrows, meaning this reaction can go from glucose to glucose 6-phosphate, but it can also go backwards from glucose 6-phosphate to glucose. So this is an amphibolic pathway. Most of the reactions in aerobic respiration are amphibolic pathways, meaning they can proceed both ways. I don't know why they have it drawn this way, probably because it's split into three, but it can move this way, which it normally does, but you could take glyceraldehyde 3-phosphate and make fructose 1,6-phosphate, and that's why the arrow is going that way. So this is an amphibolic pathway. Breaking down lipids into glycerol and fatty acids is also an amphibolic pathway, and you can see that the lipid has an arrow going this way into fatty acids, but the fatty acids do have an arrow going that way to make the lipid. So this is an amphibolic pathway also. Anyways, my point is, those of you who eat a high-fat diet, if you are a couch potato like me, most of the time you will break down the lipids, make acetyl coenzyme A or glycerol, which will be converted into that molecule, dihydroxyacetone, and then you'll make glucose until you need the glucose to perform aerobic respiration, and then you will use glucose to make aerobic respiration to make ATP in the normal way. That happens the same with carbohydrates and with amino acids. A marathon runner can digest lipids, meaning fats, directly by aerobic respiration. However, the marathon runner has to be 45 minutes into their run before they start digesting lipids. For the first 45 minutes of their run, they will be making glucose backwards and then sending glucose through the reaction, or they will be digesting carbohydrates and glucose and breaking them down into glucose and then sending it through the normal run. My point is, the marathon runner has to be running for 45 minutes before they will start digesting lipids in aerobic respiration, meaning they'll break it down into glycerol, which will come here in the glycolysis, and lipids, which will come here in the Krebs cycle. Any question about any of that? All right. I think I'm going to stop here. And we have 2 more slides, I think, of this lesson, which isn't bad. Well, 3 more slides. Any questions about 4 more slides? Is that right? Any question about amphibolic pathways or aerobic respiration? If not, I will stop here and I'll see you next time, unless you're in the 7 p.m. lab.
Thank you.
All
Thank
right.
you.
You're welcome.